Okay, I think we're going to get started. And for those of you who are brave enough to come out in this storm, you're going to be well rewarded, because I know you'll have a wonderful hour ahead of you, because Alonso and I worked together for 11 years in the same research organization. So we've been close colleagues since we were practically children. Right? So, Alonso Aguirre is an amazing person. He was born in Baja California in Mexico, and he was someone who was so inspired by the sea. He was always fascinated. He was fascinated by animals, particularly marine mammals, and he actually graduated from high school when he was about 12 or 13, and no, seriously. And then he went to veterinary school, got a veterinary degree, but he was still a child, so he couldn't practice. So his father sent him to Canada to learn English, and he went on to get a master's degree and a doctorate in wildlife management and wildlife epidemiology. So he has you know, like many of our Stony Brook students, he's the first in his family to go to college, but he has degrees for everybody in his family to pass around. He has worked all over the world. He's worked in Hawaii for the National Marine Fisheries Service, where he discovered the disease of the world's most endangered pinniped, the blue-eye disease, in the Hawaiian monk seal. He's also worked on foxes in Sweden. Well, he's just worked on so many species all over the world, including sea turtles in Peconic Bay. He has been, or I think he's currently, the president of the World Wildlife Veterinary Association. I probably mangled that. He's also an adjunct professor at Tufts University School of Veterinary Medicine, and he's also an adjunct professor at Columbia University, where he teaches a course in disease ecology. He is now the senior vice president at Wildlife Trust, in charge of the conservation medicine program, and he was the senior editor of the book that defines the field of conservation medicine, and in fact he is actively producing the updated volume, which will be appearing in a couple of years. He just told me the first chapters come in. So he's also written a book I'm sure you're all familiar with, Helminths of the World. I'm just joking, because not a lot of people study worms in great detail, but if you have a question about that, he's your man. He also is a fantastic guitar player and an inspiring dancer, so there's really nothing Alonso can't do. But I don't think you'll see that side tonight. Tonight's Marine Mammals as Sentinels of Ocean Health.
Thank you, Mary. It's really a pleasure to be here. As I was putting these slides together, I wasn't sure if she put those slides for me before, or because we have worked together for several years. And so it's very strange to come and lecture about Wildlife Trust when she actually founded Wildlife Trust, and although we are about in the field of conservation medicine. I'm bending because the microphone is too short, and I don't know if I can move it out of the floor. Mary asked me to talk to you about marine mammals because that's an area that we have worked for several years. So basically, I'm going to tell you what Wildlife Trust is all about, very basic. We're trying to empower local conservation scientists worldwide and really try to provide scientific information, very innovative, that's culturally appropriate and very cost-effective in trying to solve problems at the interface of human and wildlife conflict. It could be diseases, could be, for example, elephants raiding crops on people's homes, or could be frogs going extinct because of fungus. We try to be there on the ground with the local people. At the end, trying to protect human health and wildlife health for a long-term sustainable way. We try to do that with several programs. One of the center parts of those programs is Wildlife Trust Alliance. You'll see Mary here just above me on the left side of the slide on a meeting that we held in Venezuela. But really it's bringing these local leaders with local funding trying to solve global problems. Not too many NGOs focus in conservation medicine, in conservation of endangered species and ecosystems. We're a very relatively small organization, maybe 22 in staff in New York, 5 in Florida. But the hallmark is really partnerships. And really, Mary was extremely successful in creating these alliances with universities, NGOs, government agencies to create very exciting programs. For example, at the bottom, we have our relationship with CERC, the Center for Environmental Research and Conservation, that actually Dr. Lan Melnick, present here, her husband created at Columbia, and it's a very wonderful collaboration because that provides Columbia for free teachers, not sure that we get paid, but a really interactive interdisciplinary program in bringing several fields from botany, the American Museum of Natural History, WCS, and Wildlife Trust into enrich really the environment for many students there. We have the Consortium for Conservation Medicine, several agencies and public health schools are part of it. And finally, a national program that I'll tell you very briefly about it, working with aquatic species, mostly 2 species, manatees. As you can see in this slide, these are not fish or tadpoles. These are actually manatees congregating in one of the water, power plants, the nuclear plants that the government is trying to shut down. But we won't know the effect of these plants being shut down on the health and long-term survivability of manatees. Why? Because they depend from these warm water springs during the winter. They need warm water temperatures to survive. As you many know, a manatee just showed up in Massachusetts in recent months. Very unusual migration. They've been shown up in the Carolinas as long as probably New Jersey, but nothing like having this manatee in Woods Hole almost. So the manatee was brought by a truck immediately back to Florida and died in the way. Probably a very, very wrong way to manage an animal. That animal should be put on emergency care in a captive facility right there in New England Aquarium probably, and deal with it and the health issues and the stress issues and the emaciation and starvation process that was happening. But anyway, to make the story short, we work with manatees and right whales. In a way, this visionary perspective also brought by Mary in bringing the locals in to be part in an egalitarian network of scientists in trying to provide, for example, work with the locals in growing rainforest trees and plant conservation corridors in Brazil. We do that through our partner, IP, that has worked with the landless people that has received land from the government after land has been fragmented, and they're actually trying to learn about new forest techniques and still trying to protect the few fragments left of the probably 1% to 2% forest, Atlantic forest for the whole state of Sao Paulo. So really, and rediscovering our logo, that's the Black Lion Tamarin, about 20 years ago, and now working to expand their populations. So we know everything is interconnected in many ways. Obviously, we have to be very culturally sensitive, trying to understand culture and how we relate to the locals. We have to work on the front lines around the globe and trying to enhance health of people, ecosystems, and wildlife. And at the end, we are the only NGO focused in conservation medicine. The whole thing started in the mid-90s, really, and way before that, probably Michael Soule came up with the concept of conservation biology, a crisis discipline. I remember when I was in graduate school, when I was taking ecological geography, from one year to the next, Professor Benke decides to change the name of the lecture to conservation biology. And so I said, what are we trying to do? I thought we were trying to learn the distribution of and relative abundance of species, basically ecological factors of natural populations. And he said, well, actually, we're counting species to extinction. So I think it will be very wise to call my course Conservation Biology, because that's what we are trying to do, to identify factors that are leading species to extinction. We haven't changed in 20 years. We haven't planted enough trees, and we're still trashing the planet. In fact, at these rates, we need 6 planets to sustain the current population. How are we going to handle it? Just coming back from India, it's devastating to see the level of pollution, number of people just concentrating in that part of the world. And yet we in America, or North America, we consume 1/3 of the goods of the planet and produce 2/3 of the trash. So it's a total imbalanced situation. Ecosystem health has come as a term for a while. There's 2 books on ecosystem health. The problem with the term we had at the time is very anthropocentric. We're looking at human health, community health, then ecosystem health. What we decided then is move ecosystem health to the side with human health and biotic health. So the interaction of these 3 circles right here is what we're calling ecological health or ecohealth. In fact, Marin's vision of creating a program here on Ecohealth in the next future, that's where we're going on One Health. Really, that health connects all species at all levels. So you can get this in Amazon.com for a small fee, used for half the price. No. It's a very good book. Actually, this is the first and only book on the topic, and I'm working on the second one. Hopefully, will come out next year. Mary doesn't know, but she may have to co-author a chapter, so it'll be coming her way soon. Well, basically, it's defining ecological health in practice, and that's what conservation medicine is all about. And in 2003, EHP, Environmental Health Perspectives, the magazine of NIH, produced an article linking these 3 circles. And it was a break for us because that's when the public health medical schools realized that conservation medicine and emerging science and the need of medics, veterinarians, ecologists, sociologists, modelers, politicians, local people work together to solve the environmental problems that we face today. And we have a journal. I'm a co-editor. We have an association, the International Association for Ecology and Health, or International Association of Ecohealth. Jonathan Patz is the president. We're part of the board. We have this magazine now in its 5th year. You can probably receive it through your college, I'm sure, for free, ecohealth.net. An article came that I just wrote just came out, and so you have to get it. It's an interesting magazine because we have survived. This is our new logo for International Association for Ecology and Health. We have impact factor 2.3. In 5 years, I think it's amazing. It's very excellent. And one of the things people like is the holistic approach to problems. So we're bringing health, ecology, climate change, socio-cultural problems into the table and practical solutions to some of these. Please subscribe and submit your work today. You may enjoy it. So now getting to conservation medicine, one big issue we work with is combating global emerging diseases. The main issue of the world, Mexico City from Popocatépetl at night, this is the core issue. We're too many. No other species in the planet has grown geometrically in this way. You can see a little bump here due to the Black Death, but then we recover extremely fast. By 1998, we're 6 billion. We are reaching 7 billion today, and apparently we'll reach carrying capacity at 10 billion. Now, I told you we already need 6 planets to maintain 6 billion. So probably we'll need 10 planets by the time we reach 10, right? And 12% of the the US population concentrates right here. And if you can see New York at night, we are someplace around here, I should say. Still nice, not too many lights. But yet you have 4 species of sea turtles, lots of marine mammals coming in here, black bears, wild turkeys, white-tailed deer, among other wildlife, and people are totally disconnected to their environment. You still find New Yorkers and kids that think that milk and eggs come from a supermarket. In fact, we have chickens. Laura was surprised. You have chickens in New York? I say, well, a little bit outside of New York, I will say here. But still, you can raise some chickens. And still, you can have a way to really integrate the human ecological impact with what we have left for granted. And that's that wilderness, that contact with nature, what contact with what we have around us that we still enjoy. And globalization. The Global Aviation Network published in a paper back in 2004 of number of passengers traveling in one day in 500 airports in 100 cities. So we have 10 people to 25,000 people traveling in one day. Basically, you can be in any part of the world within 24 hours. And that has caused what we call pathogen pollution, movement of pathogens from one area to the other in the planet. And some of the hallmarks that we have worked with is, for example, look at, identify the trends of Nipah virus, look at SARS, look at West Nile virus in New York. So we actually trying to identify programs in the hotspot regions. We have predicted the entry of avian flu in the past. And it won't come from migratory birds. It will come from poultry and humans, illegal or legal markets. And we have created the hotspots of emerging infectious diseases. In fact, USAID, an agency from the government, has released a big proposal based on these hotspots. And I'm talking map A, where hotspots were identified just 1 year before. You see Mexico, and boom, we have H1N1 showing up. Shall we blame the pigs for H1N1 when the actual virus was identified having 2 human strains, 1 wild bird strain and 1 pig strain? Still, we're struggling with the origin of this outbreak, but we saw in Mexico City and then the rest of the world to be aware of a disease that can be lethal and fatal. And it's not only our experience with H1N1, we have in the past H5N1, SARS, and West Nile. This is the Google map that came out just 2 weeks after the first case identified in Mexico City. So you can see how fast disease can travel around the planet just by globalization, by movement of planes. This will be the fastest way, but probably, hopefully, pigs won't fly. The concern is if H1N1's transmissibility combines with H5N1 pathogenicity, we'll have a very hot virus that may kill 400 million people. And that's the big concern about these 2 diseases. If in Bangladesh the true strains exist, or Thailand, or other Southeast countries in Asia, will show up a recombination of H5N1 and H1N1, then we'll have a very hot virus, easily transmissible, and that can kill people just by direct contact. And that's the concern of these pathogens. We don't know how they're going to reassort themselves, and we're hoping for, we're expecting a very hot H1N1 season this winter. So you have to be careful on dealing with that. But you came here to hear about marine mammals. I was told I have a whole hour to talk, and I don't like to talk too much, so we'll try to get you where you wanna see. Before I hit on the mammal issues, the oceans are in trouble. In high school, I remember the geography teacher would say that the future of humanity was the oceans, that there's no way possible that the oceans could be exhausted in their resources with human population, and so we can use all these resources as far as we can go. How wrong she was that within half of my lifetime I have seen oceans depleted all around the planet. Many people depend on these resources, and yet we're removing way above carrying capacity. For example, why they tell you shrimp is wrong to eat? Well, it depends. Not all shrimp is wrong to consume, but if it comes from specific fisheries in the Gulf of Mexico, you may be capturing 12 pounds of juvenile fish killed by 1 pound of shrimp. And that happens with 12 pounds of juvenile fish is just dumped with no use whatsoever. So about 80% of fish stocks really require action. And the problems are that too many fleets, massive subsidies, fishing pressure. We are totally unsustainable. The statistics that fisheries people give you, National Marine Fisheries Service, that loved in this part of the country, and I worked for them in the past. I know it's a total conflict of interest that one organization is in charge of putting fish on the American people's table and yet conserve marine mammals. See, it doesn't make sense to me. It's totally against everybody's interest. So here we're trying to conserve monk seals, and the populations are collapsing because of lack of food, but then we're overfishing and trying to get as many fish as we can as possible using the agency's philosophy. And the philosophy shows right here. I don't know how many of you remember The Perfect Storm, the movie, a few years back, when this fleet went to Georgia's Bank, and the fishery, why? Because there were no more cod in Cape Cod. So I had to go all the way to Georgia's Bank and to a storm, and they all got killed. But then again, this collapse in cod within northeast US hasn't been recovered since then. So we still have the collapse, so we have to import cod from other parts of the world. Another issue is that we have very few protected areas. Probably 10 to 30% of the Earth's landmass is protected as national parks, wilderness areas, even biosphere reserves. But only 0.25% of marine habitats are protected. And that includes coastal areas, coral reefs, the seafloor. And we know so little about these species that we know the extinction of 4 snails, 5 bird species, and 3 marine mammals, including the Mediterranean monk seal, that was the most recent extinction, and the stellar sea cow. That's so little we know about those ecosystems. And the core problem right now in the oceans is this, that we're really fishing trophic levels. I remember what my grandmother considered trash fish and fed it to the pigs. Now it's prime fish in our tables. Why? Because there's no more of these big guys. In fact, 90% of all these big predators are gone in all the oceans. 90%. And as you know, the Japanese are willing to pay $2,000 for a pound of bluefin tuna. But to find a bluefin tuna now in the ocean is very difficult. And so the issue is that we alter an ecosystem structure and function, and so we are not able to cope with the system. Yes, we have some ways to mitigate that by creating these open ocean pens that are becoming now very prolific in several parts of the world. This is a huge pen, and you can see fish just swimming around this basic metal structure and being fed. But what we know is, or what we don't know is the interaction of these fish, the stress caused on these fish populations in captivity, and the diseases that can be moved from those pens into the ocean. We know very little about it. We know about the farm salmon and spilling diseases into wild salmon. Chinook salmon, for example, in Washington, have acquired a couple diseases from captive salmon. And so we know that those interactions are happening already. On top of that, we had climate change. We were just having a lovely dinner by the windmill. Beautiful. Actually, when I came out of it, I looked at it as it's a real windmill. I didn't realize it reminded me of Don Quixote de la Mancha. I said, oh, that would be great to have a show there. But I figure I'm too old to do that. So anyway, 35% of 1,000 animal and plant species in 6 regions are at risk of extinction from climate change. And that's work from Ruth and Thomas in Nature and Science, about 5 years ago published. Those numbers are changing, obviously. But I put a polar bear because remind me that the weight of polar bears in recent surveys had dropped 20%. So bears are 20% lighter than 10 years ago. Why? Because they don't have prey to feed on. What happens with walrus? Now, walrus, being a pagophilic species, has switched prey. And now they're feeding in ring seals. And you know that walrus are consumed by Inuits or by some Eskimo populations. They can legally hunt them and consume them for their own purposes. So what happened with the switch in prey, we found out that ring seals are infected with trichinella. And trichinella is a parasite of pigs. It's Tenia. I'm sorry, it's together with cysticercosis, but trichinella is a parasite that insists in the muscle of pigs and circulates with pigs. Well, that was introduced into the wild a while back into polar bears. We know that bears have it, probably feeding into ring seals, that's the cycle. But now walruses are infected with trichinella. What's going to happen now, the Inuit are going to consume walruses, and we have a zoonotic problem. We'll have now humans getting sick of trichinellosis. That's basically lethal disease for humans. So you start seeing all these changes in prey availability. And a big question with marine mammals. We've been measuring contaminants for years. In fact, ecotoxicology is a very important area that we've been trying to develop at Wildlife Trust with Mary's initiative, with many people, because we know, for example, in recent studies, newborn in Philadelphia, actually this group, environmental group, picked up 10 newborns in Philadelphia downtown, took a blood sample as soon as they were born, sent a sample to the laboratory, and they measured 287 different chemicals, from plastics to DDT, that's supposed to be totally gone since '71, to PCBs, to dioxins. So 287 substances found in those newborns. From those, 150 are known carcinogens. They cause cancer. So who wants to measure the blood levels? I don't. Or if I like raw fish, I have enough mercury probably. But you start looking at these levels, what do they mean? When marine mammals can harbor 100 to 1,000 times the levels of contaminants that other species can. So we just start to explore these factors. And maybe this, if we add a stressful situation, actually it's a photo taken in one of the most pristine areas in the planet, French Frigate Shoals. And you have that turtle, drinking a bowl of chloride and then after a bowl of cognac. No, actually, this is the garbage being collected by National Marine Fisheries Service personnel every year. When I started in the program back in '96, we used to collect about 80 tons of garbage every summer. Now I send a student, Joe Spring, to document, and I was so hopeful to be collecting half of that. Well, no, it was twice as much, just 150 tons of garbage being collected. And yet, where the garbage comes from? The garbage patch, as you may know. We can document marine mammals' direct effects, human effects, by this boat in Florida going through a dolphin's fin. Easy to document. Or this sea lion going through a fisherman's net. And on top of that, the net is here, but the animal has been shot. At an X-ray, you can see the bullets. So it's really hard to measure the interaction at those levels. The effects of contaminants are interesting too, because you have the acute effects. And this is an old photo from Colorado, Wilson's phalarops being poisoned with eltrine. And this is a substance, an organochloride, that inhibits acetylcholinesterase, that inhibits the transmission of the nerve impulse. Or you have the chronic effects of DDT, still seen today, despite the ban back in 1971. We still promote production. Why? To control malaria in Africa and India, for example. So we wonder how diseases were transmitted from terrestrial animals to marine species. Easily, you know, just a dog brought up to a seal population. Maybe this disease, like kind distemper, can jump into this population. Well, in fact, that's what happened. And that's what we think happened. That this, there was an outbreak of canine distemper, and these animals were thrown into the North Sea. And one day to the next, or a week later, we have 20,000 seals dead by canine distemper. Also, we have the famous disease harbored by cats called Toxoplasma. Toxoplasma gondii is a parasite that your grandmother warned you if you had a cat and you're pregnant. Be careful with your cat because you may abort or your kid may get encephalitis or blindness. In fact, they were partially right until cat litter was dumped into the ocean through toilets in California. And now we have sea otters dying of encephalitis because of Toxoplasma. This is a classic example of pathogen pollution, of having a parasite finding a new host and saying, oh, this is great. But then you gotta be careful because as a parasite, you gotta make sure that your host survives. So that's different issues. Changing in prey of sea otters have increased their susceptibility to other parasites, like Polymorphus. Polymorphus is a parasite that circulates between gulls and sand crabs. How did the sea otters get it? By feeding sand crabs. Well, wait a minute. Sea otters don't feed in sand crabs. They feed in abalone and sea urchins, where there's no more abalone and sea urchins. So you gotta grab whatever they have handy. So they picked up this parasite that causes severe hemorrhagic peritonitis. So now enteritis, peritonitis, and now the animals are dying also of a new parasite. Algal blooms have been detected recently more and more often. We have several groups, the dinoflagellates, and probably I like to mention brevet toxins for manatees, and we'll talk briefly about it. Seawater toxins for monk seals, domoic acid for sea lions, and saxitoxins for some other species. But really have occurrence and distribution have increased. Looking at recent data, indeed it has increased. Not only because there are more observations, or people have recorded most of these events, but we have changed the ecology of many of these environments, increasing these algal blooms globally. And a new paper by Frances Gulen and her group in 2007 also has seen an increase of marine mortality events. A lot more marine mammals are dying of diseases from the 70s if we compare to recent years. So domoic acid basically makes holes in your brain. And this is hard to see probably, but you can see this pale area. And so you see these sea lions like twitching to the side with central nervous system, an attempt to rehab. There was a big mortality event, about 2,000 sea lions dying of domoic acid that they acquired by eating herring. And what happens is when you try to rehab them, it will work out to a point, but as soon as they will have a baby, a pup, they'll kill it. Their behavior is totally anti-maternal. They won't go for it, and most of these animals will die over time. So rehab efforts are totally useless in these situations. But yet we have toxins like brevet toxins, and you can see from the 70s that have been manatees, look very carefully in Florida. In recent years, starting in '96, we have severe mortality. In 1997, we thought we were dealing with morbilli when almost 200 manatees were killed by this brevet toxin. And so as we add several factors in a population, you're going to stress the hell out of it. And of many individuals, you're going to stress and cause a different situation in their environment, and that's going to make them more susceptible to disease. And we thought manatees wouldn't get sick of a disease and until we find now a new papillomavirus. You start seeing lesions in manatees after brevet toxins are known to cause immune suppression, and now these new diseases are showing up. And during this presence of diseases, we start looking at what people are doing with these wonderful trees. And I don't love the genetic trees of animals, but we look at the virus level, and you start looking at morbillivirus, for example. And we have, for example, this is nucleotide differences. And you start, let's look at this group, measles and rinderpest. Rinderpest is a disease of cattle. One of the first one, actually the first one, almost manifested way back then, very close to measles in humans. So you start thinking, what happened here? What happened is we domesticated cattle, we got exposed to their virus, or maybe cattle got exposed to our virus, and they developed their own strain. So if you look at that data very carefully, you start looking, well, and then we have a close virus, Peste de Petit Ruminant. That's a disease of goats, similar to that. But then we have canine distemper, the disease of dogs, where we thought lions wouldn't get it. That's how I was taught at school. Back in vet school, they said, well, canine distemper is a dog disease. No way cats can get sick. And then we have that canine distemper kill 90% of Serengeti lions in 1996. And they acquired the disease through domesticated dogs from the Zulu tribes around the area. So these are very close. Canine distemper kills seals. Phocine distemper switches to their own pathogen within seals. So we have a new virus. And then all of a sudden, we have on the other end dolphin and porpoise distemper, or morbillivirus. All of a sudden, we have 5 new viruses killing thousands of marine mammals in the past 15 years. We're talking about thousands of animals. And that's where we came with the concept of sentinel species. Can these species tell us something about the environment? I had to put a sea turtle in there. This turtle is suffering of phyropapilloma. Is related to biotoxins, to a virus, to stress populations, to warm water temperatures. All the factors that we've been seeing in other populations, you can see on these turtles. What I really want is that these sentinel species will tell us those multiple stressors, how they accumulate in the environment, how they're synergistic, and can they warn us about what's happening before time. And the way we do is we look at individual health, we collect data, population health and population biology, and that will be part of the puzzle for our ecosystem. So it depends on your question you're going to answer. That's going to solve your impact, for example. And if we can measure that impact with a population, maybe that will tell us a little bit of that system. And that's what we're trying to do here in Peconic Bay with Riverhead. Rob De Giovanni is here. And we have worked for 5 years with sea turtles in looking at the health when they come into Peconic Bay and when they depart. So we're looking at trying to gather information if are they picking any contaminants, any diseases. And really it's an opportunity to integrate our scientific knowledge in the marine environment and transmit it to the public, stakeholders, the children, and the decision makers. Really, that's a very good way to be in contact with them. So more in detail, I'm going to tell you about 2 examples, maybe 3, and then I'll wrap up on how these new diseases are coming into marine mammals. We know that natural mortality is normal. Many biologists struggle for years. We support that disease is part of populations that gonna kill animals and they will recover. They not necessarily be density dependent, but density independent. The problem now with globalization, with human interaction, we're seeing large-scale threats like morbillivirus and biotoxins. We see many cardiovascular problems, many lung problems, and many of these strandings are totally, sometimes consistent and predictable, and sometimes evident. But we know the emerging diseases are more prevalent. For example, in cetaceans, strandings there may not be as evident in pelagic species. But now, in recent years, we've seen more pelagics stranding. And pelagic means these animals that are farther away from the coast, not like tursiops or like other species. So we looked at national marine fisheries data. As in any wildlife disease, 50% of cases are undiagnosed. We don't know what happens because the seal is too rotten, the whale is turned into a soup, so there's no way to collect samples. We know maybe the species, and that's it. But in recent years, 1/4 of that mortality is caused by morbilli, and almost the other 4 by HAVs, algal blooms. So those are two major causes of mortality, most likely linked to human impact. So if we look at cetacean strandings, they are difficult to judge. It took me 3 years to get permission from the same agency so I could get a blood sample from a monk seal, because stress supposed to be a disturbance, human disturbance, was the main cause of on trying to extirpate the species. So after we were able to get a blood sample, because we're not magicians, we need to know what's happening with that physiology of that animal. We can see a very healthy dolphin swimming nicely and then found dead next morning. And that's very common. So we know that we need a blood sample. We know that coastal species usually strand singly only when they're ill. We know nasitrema, that's a parasite, will cause terminal brain damage. And many pelagics will strand in good health. At the end of the day, we don't know what's the survival of those animals we released. Until recent times, we're setting up some radio tags, satellite tags, that will look at these animals. And finally, large whales, most of the cases we have to euthanize. It's very hard to rehab a large whale. Returning small cetaceans, as Rob may tell you, is extremely difficult, very costly, extremely time-consuming, and yields, in general, poor results. So I'm just telling you these things as we see a perspective on rehabilitation of these animals and what's going to be their future. Every cetacean has parasites, as every pinniped. You'll find worms everywhere. In fact, severe lesions loaded with these parasites like this crassicauda, you'll see in the blubber of every cetacean pretty much. What's the impact? We don't know. So we have to be proactive. We have to actually go out there, capture these animals. You see a dolphin here with Randy Wells' group in Florida, already radio tagged, ready to be released. And basically, 3 generations of dolphins have been sampled and very well known in Sarasota, Florida. So that's the type of studies that we need. Why? Because we get baseline information, we can see the impact of disease, we can have only the long-term monitoring is the only way to measure success, really. And that's taking us a while. Why do we need to apply these techniques? Is, again, rehab, response to morbidity, mortality, maybe treatment. People ask about treatments. We were talking about the species like this will strand. If you're lucky, you'll get a whale like this. Most of the time, I can tell you it happened in Denmark. This biologist was getting close to this whale, and the whale blew up because it was totally full in gas. And then you end up with chunks of blubber all over your face, all over your body. And it's a very stinky experience. I don't recommend it. And greasy, too. Then again, you have a massive stranding. What do you do with these animals? Many of them are alive. Are you going to release them back into the wild? How many shall we sample in these massive strandings? We have a few dying. We have some healthy ones we can move immediately back into the wild or get a blood sample. So we raise all those questions when we deal with these strandings. This photo was taken in Hawaii by a good friend, George Balas, and released then later on as a postcard. And these two species are very close to my heart. I worked with both of them for several years and trying to identify the cause of their decline. And so we looked at many factors of affecting, for example, Hawaiian monk seals, from environmental factors, disturbance, disease. Host factors could be nutrition, as I said, genetic, hormonal, and also solar radiation. Monk seals live in the northwestern Hawaiian Islands. When I started in the program, we used to have 2,100 seals, about. It's the most counted pinniped in the planet. And we know pretty much how many seals are there. Now we have about 1,200. So in 10 years, that's about 40% decline. At this rate, probably by 2020, the species will be extinct. And that's what's expected. This is the largest population, and we move along these islands, the northwestern Hawaiian Islands. And so we see a decline up to 2,000. Now we're about 200 here. The causes of decline are multiple. We'll see mobbing, for example. We have here 6 males trying to mount a female until they kill it. And that's called mobbing. We see shark predation at all levels. And we see, obviously, entanglement. Entanglement is a major issue with Hawaiian monk seals. And this is the debris, for example, that's collected in those islands every year, 150 tons of there. So back in the 80s, they decided to start a rehab program. Let's bring 6 females into captivity after winning, fatten them up, and 6 months later, we release them back into the wild. So we help them go through that hump of lack of survival during the juvenile months. So seals will bring into captivity. That year, in 1995, that's when I got hired, we brought 12 seals. We got very cocky. We were doing very good with 6. So that year we brought 12 female seals. You know why females? Because of mobbing. We don't want to rehab males at this point. They were brought into Oahu, then screened, force-fed, free-fed, and then transported back into the wild. And in 95, when I got hired, we got this blue eye disease, as we call it. Probably a herpes. Hasn't been proven today. You want to see these seals, you can go to SeaWorld San Antonio. You still see 9 of them. 3 of them died already. And first they started squinting. They started with photophobia, conjunctivitis. 3 months later, they showed the blue eye. And a year later, they all went blind with cataracts. And so you start to see here the lesions, the blue eye, the squinting, the inflammation. We have to dilate the pupil to show this cataract. You see here the slit here, probably not too good. And then the whole cataract, full-grown cataract. And so if you look at each seal, you start seeing that's an infectious process. Most of these seals, for example, this one, develop blue eye but never cataracts. In blue, we have the seals with cataracts. This one stopped here because it died of another condition, the same with this. But we have this seal, this is our golden seal, never developed any clinical signs. But we cannot release it because he got exposed to other seals. Until we don't know what are the factors of this disease in seals, we cannot really act properly. The fate of the population is unknown. Really, to this date, after I left the program back in 98, to join Wildlife Trust, and we have published a few papers after that, I think the future doesn't look too good because we still don't understand the factors of decline. We still have National Fisheries Service increasing quota for fishing, and yet not protecting the resources as they should. I'm going to tell you Baja because first I was born there, It's a very productive ecosystem, highly endemic. 80% of the marine mammals in the Pacific come there. And it's relatively pristine, but you'll see the changes. This is my last example, and we can open the sessions for questions. We have the most endangered. Mexico is qualified to have the most endangered, the most corrupted, the most everything. Actually, no, it's not such a bad country. After being in India, I'm not sure if it's the most corrupted country in the world. But it is up there. We have our environmental problems. We have a lot of concern for the environment. That's the only country that has protected all their shoreline for marine mammals by law and has really done interesting programs. But we have La Vaquita. I tell Lorenzo Rojas that runs the program, he says there's 600. I tell him that's extinct. We haven't seen one probably in 2 years. The speculation is probably less than 100, probably already ecologically extinct. And that lives in the Alto Golfo, right way up here, way up here. So, and I'll show you a map. We have the Guadalupe fur seal. What do we know about Guadalupe fur seals? Who in this audience know that there was a Guadalupe fur seal? And this is the way they rest. So there's a documentation of the species. We would like to start a project to start capturing and looking at behavior, ecology, and health of these animals, probably in the 10,000. And we have islands, 267 islands in the Sea of Cortez that Ed Ricketts explored way back in the 60s, and totally probably he'll be shocked, in the 40s, I'm sorry, totally be shocked with what's happening there now. But we have islands like Isla Raza that will host 2 species of birds. One migrates to the North Pole, the other one to the South Pole. And 95% of those 2 bird species nest in this little tiny island. And you wonder why. Why? Because we have a massive production of resources. But we start seeing the impacts. We start seeing the bycatch of sea turtles and sharks. And you can see this by Norbert Wu. This is not Baja, but this is the type of situations you start seeing. You see the border that Bush wanted to build through Mexico, and we already have it in Tijuana. It's a total disconnector. And then the development of these areas. This is the vision of the Protected Area Program. This is the vision of the Tourist Department. So yes, we want a road across the most protected area, right here by the Los Sirios, where we have cacti that take 300 years in reproducing, unique to these ecosystems. In fact, Baja gives Mexico #1 in cactus biodiversity because of the number of species found there. So they want airports, they want golf courses, hotels, but there's no water. So what are we going to do? What we start finding is that these areas, each one of them, are important feeding areas for sea turtles, for example. Or gray whales. That's where they reproduce. That's where they spend the winter mating and reproducing in San Ignacio mostly, and coming back in the summer months to Alaska. In fact, these animals have been killed before. This is a small whale being collected samples. The animal died, so they're taking blubber samples. And what happened here back in the monitoring times, you see mortality is very low. Up to 1999, we found almost 300 females along the range stranded. In 2000, about 400 males stranded. So we concluded that this mortality was related because we have too many whales, and there's not enough food to keep them. That was the conclusion of National Marine Fisheries Service. When indeed we look at the distribution in their aerial surveys, we start seeing mortality strandings all along, but mostly here in the breeding grounds. Something's happening here that we don't know what it is. We found very machated whales, very skinny whales. And by 2001, we just had 20 gray whale strandings in the whole range. So from 400 drops to 20. We go back to normal, we declare this is over, we don't determine the cause. But we only were able to do a necropsy of 2 whales. And one was with Eastern equencephalitis, the other one with biotoxins, domoic acid. What's the cause of mortality? We don't know. But probably we have too many whales. So let's have the Mauca tribes now start hunting whales and go back into that issue of consuming wildlife. But in fact, in recent years, we found a morbilli in 2 calves and a female. These are samples collected from 3 strandings and several tissues, and you see reaction to morbilli virus. Similar to the dolphin morbilli. If morbilli gets into the gray whale population, that will be devastating for this population and probably a cause of extinction. My last example deals with California sea lions again. In fact, we think that may be a different subspecies. We looked at northern Gulf of California. We have several populations in these islands, and we have collected samples since 2004. We go to the colonies, we capture the animals. This is just numbers found by individuals. And if you start seeing a trend since 2000, there's a decline. You start seeing numbers rapidly declining. You can see the trends of population pop production at several islands. This needs to be updated, but we are around here. And so we see that production has been declining too. And everything that we don't know, we blame to El Niño. El Niño is the cause of any other problems in wildlife because we cannot figure out what's happening. And so we see some correlations to El Niño, a little increase, a major decrease in 2006, and it hasn't recovered since. And so we go out there, we anesthetize the animals, we use a net to capture them, we get blood samples, we get all kinds of samples to really take care of these animals and try to identify causes of mortality. We weigh them, measure them as a good biologist does. And we start seeing that the condition index of these populations is declining too. This is based on a publication by Castro Gonzalez, where we see several populations, 2004, 5, and 6, and indeed the condition of the populations has declined too. And not only that, a paper published by Bowen in 2005 has shown an MH3 genotype related to cancer. There is a herpes virus that causes sarcoma in sea lions. I'm sorry, a carcinoma. This carcinoma is killing sea lions in California. And so we're trying to figure out this carcinoma can affect humans. So Francis Gulan got a big grant from NSF and NIH to look at this. And they found an MH genotype and cancer specific to this gene found in animals with cancer. And this gene is linked to cancer. So we know that animals with this gene are at least 3.3 times more susceptible to cancer than animals without it. So looking at positive samples, you start seeing that animals also further looking carefully positive to the herpes present this gene that makes them susceptible to this specific cancer. So in a study, we looked at populations in Baja of sea lions, and we start looking that several of these populations are highly susceptible to this cancer and to this herpes, even though we haven't really identified this cancer. They have the genotype, so we just need the right environmental conditions to change the behavior, to stress out the populations, and probably we start seeing animals with cancer. And so basically, just to finish, we know that these populations, as we stress them, as we concentrate them into smaller areas, are going to suffer more diseases, more interactions with us. I was sent to the US many years ago to get a degree as a zoo veterinarian, and I refused. I said, no way I want to work with captive populations. Why? Because captive populations are feeding The wrong nutrition is just a fake nutrition, it's not natural. Captive populations are concentrated in an initial way with many other species. And they're suffering of malnutrition and new pathogens. And also they get exposed to contaminants like lead in the cages back then. Well, I come to realize after those 20 years in working with these populations, I'm not a zoo veterinarian, I'm a mega zoo veterinarian. Because we're working with these populations in very confinement, in real confinement. We're not letting enough space for them to roam healthy and stress-free. And so we're looking at health as a powerful motivator. And I just put these slides to remind you West Nile virus. Really, that's when people from New York to California woke up and said, well, maybe we have to do something for our environment. We need to really work on surveillance, change research into policy. We're working really hard on that. Keep educating the public, changing our attitudes towards those species, impacts in ecosystems, involve the community. I'm sure you guys are very active on that, and trying to achieve that balance. I don't want, as we started the Conservation Medicine book preface, saying that the children of the third world will explain about climate change, meaning that the few humans remain on this planet move to a different planet. Since we have lots of planets out there, maybe that's the only option. Anyway, we need to cross borders, work with many cultures, with many teams, with many disciplines in trying to achieve a balance. Thank you for your attention. Yes.
May I?
Yes, please. I'm sorry, yes.
Along
yes.
with your measurements about the incidences of various morbidity were you also tracking changes in the pH of the ocean, temperature ranges over those periods of times? Is the chemistry of the ocean changing?
Absolutely. The question is if we have measured some pathogens or some biotoxins. Has the chemistry, physics of the ocean has changed? Yes. Not only that, but physical characteristics like temperature. Now we see warmer waters in some areas. We know that warmer water temperatures have been correlated to tumors in sea turtles, to agricultural runoff. That has changed the pH and the organic content of those waters. So we see more prevalence of fibropapilloma on those waters. On the other hand, we see changes in temperature with the North Atlantic oscillation, what we call the Atlantic freshening. That means because the climate is changing, the oceans are getting colder. Why? Because the poles are melting. So those cold waters are coming way far south, and Rob can tell you more about it, but we're having more cold stunnings in sea turtles and some marine mammals. They cannot stand the cold temperatures, so they strand. And they end up here in New York, right here in Long Island. And we have seen that with Kemp's Ridley in Cape Cod. We have seen that with several species of turtles every summer or late summer, fall, because those cold water, those turtles used to roam on those areas. Now they're too cold for them to do so. So we know the chemistry and the physics of the oceans are changing. Yes?
The assistants that are assisting the veterinarians that are working with these animals, is there a specific degree that they have besides just the general LVT?
Oh, for the veterinarians working. Well, since 1992, we've been struggling if the veterinary education as a general practitioner is enough to do wildlife work. The answer is no. The veterinary college, as limited as it is and as tight as it is, just teach you to be a clinician, very narrow thinking. Even though we are real doctors, as the medics don't think so, We are the real doctors because we treat 99% of the species in the planet. Medics just treat medium to large size mammals. But really what we do need is an advanced degree in ecology, in conservation biology, zoological sciences, because that gets you the research experience, and not only that, the vision to solve field problems. And most of the time it's a team of disciplines that get us along to start identifying the cause of mortality or causes of disruption in these populations. At the end of the day, after you go into 4 years of college, 4 years of vet school, you say, I gotta go another 4 years for a grad degree. I'm sorry to say yes. And you don't get paid like a GP. We're here because we love it. Yes.
I heard that H1N1 was actually started when it was in bats and transmitted to pigs. Is that true?
Oh, Nipah virus, probably. Yeah. Nipah. It's a complex. Actually, 32 new diseases have emerged in Australia in the past 15 years. And many of them come from bats. The story is back in the 90s, mid-90s, we start seeing several cases of humans dying of encephalitis being exposed to pigs. And the story came down that bats concentrated in those piggeries in Malaysia, peninsular Malaysia, could be related to weather again, to El Nino. And they found the index farm where they saw many bats feeding on those fruit trees close to those farms. So the secretions and fruits from the bats fell into the piggeries. The pigs will feed into these fruits and get infected, dying of pneumonia, and not only that, infecting humans with encephalitis. So that year they killed over 4 million pigs and over 300 human cases got infected. In fact, SARS came most likely from bats. That's what our group concluded recently, 3 years ago, that bats are reservoirs of several pathogens, but the way we manage populations, we bring populations into contact, we unleash these new pathogens, finding new environments and new hosts. So we can talk a little bit more about it, but yes, that's an important disease of humans. In fact, recent work in Bangladesh has shown that because of the palm juice, they collect the juice from the palms and they drink it fresh. And that's the first time that the transmission has found from the juice into humans. And they're dying of Nipah too. So with camera traps, we figure out that the bats will come at night and feed on the juice, leave their secretions. Next morning, a human will drink bat saliva plus the juice and get infected. So it's an easy way to control it by putting some screens, but took a while, 2 or 3 years of research from our teams to figure that out. So yeah, many new pathogens are coming. I can tell you we know only 2% of the no viruses are pathogenic. So if we start multiplying, let's say, 20 viruses per vertebrate, and there is 5,000, at least mammals. Well, let's talk about mammals. 5,000 mammals, 20 viruses each. Just make the math. There's a lot more pathogens coming our way. So you go to vet school, you won't lose a job. You'll find a job right away. Or public health. One more question? Thank you very much for standing tonight and bearing the beautiful weather out there.